All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So there is a Swedish study that shows that the spike protein RNA from Pfizer-BionTech vaccine in vitro goes into the liver cells and is converted into DNA in their nucleus. And it also increases the expression of the reverse transcriptase line 1, which is an enzyme in all of our cells, mostly present in the dividing cells. And that enzyme is the one that helps to convert the RNA of the protein, the vaccine, spike protein vaccine RNA, into the DNA. And that DNA is found in the nucleus in vitro. So let's look at the study. It is a Swedish accepted and published study. So it's not a preprint. It is not something that is still waiting for review. It is reviewed and published. Let's look at that. The discussion that I'm going to do is in two parts. In the first part, I will give you a summary, just one-page summary. In the second part, I will discuss a high-level overview of what they found. The third part today is missing, and that is the detailed discussion of exactly how this was done. If you would love to see that, just let me know in the comments and I can do that tomorrow or day after. Okay, so let's start. First of all, the links to the study itself. So here is the first one. This is the link to the study. My homework always shows because of my highlights. So this is the MDPI, intracellular reverse transcription of Pfizer-BionTech COVID-19 mRNA vaccine, BioNTech BNT162b2 in vitro in human liver cell line. And this is researchers are from the Lund University, Sweden. And if you see here, 18 January is when they were revised. They revised this paper. 19 February 2022 is when it was accepted. 23 February was, I think, accepted and then published on 25th February. That is today. It is hot off the press. So this is the discussion. Let me now very quickly actually go over this with you. So summary. Yes, it is boring, one page, but this is the summary. So let's see. The summary is that the Pfizer-BionTech vaccine RNA, when given, so it has a lipid nanoparticle. When that nanoparticle is given in vitro to liver cells, the RNA of the vaccine is converted into DNA, which really should not happen or is not expected to happen. It's converted into the DNA, and that DNA is found in the liver cell's nucleus, again in vitro. This happens within 6 hours. It is rapidly done. And then this vaccine, lipid nanoparticles, they proved for the first time that the vaccine can easily enter the liver cells within 6 hours. Then when it goes into the liver cell, it increases the expression of a protein called line 1. And I would explain it, but imagine line 1 is a protein's name whose job is to make DNA from RNA. So presence of the vaccine itself somehow triggers in the cell, especially dividing cells, it triggers in them the production of line 1. Line 1 is a normal protein in our cells, but vaccine increases the production of that protein. The increased production of line 1 in turn converts the vaccine's RNA to DNA. And that DNA is present in the nucleus. This is the first time ever that they have shown in vitro that this can happen. The other thing that they found was these liver cells also expressed the spike proteins on their surface, or the pieces of spike protein on their surface. This is expected. Any cell that would pick up the spike protein would express it, or that would pick up the vaccine would express it. They talked about it in a different way. What they said was this expression of the spike protein on the liver cell will make these liver cell a target of the immune system and can cause autoimmune hepatitis. So this is the observations. I would explain them later, but this is the summary right now. Concerns. What are the authors' concerns? Their concerns are if the spike protein, if the spike RNA of the vaccine goes into the DNA or nucleus, becomes DNA, it can cause genomic toxicity over there. Just like when we get the virus in our body, we are in a pathological state. Similarly, when the extra DNA, abnormal DNA is found in the nucleus, that can be damaging to the remaining parts of the nucleus. So it could be genomically toxic, which in turn could become carcinogenic. They did not say that it can cause cancers. They simply said that because of the genomic toxicity, there is a possibility of carcinogenesis or cancer production, and more researches should be done. Then they said, if it can go to liver and enter the liver cells, then maybe it can also pass through the placenta. And the Pfizer's pharmacodynamic and pharmacokinetic study that was done, not study really, the distribution paper that was done in Japan, they said in that paper, they did not talk about vaccines dynamics with placenta. So they said that is another thing that should be seen. And then they said what we did not know is that DNA piece that is created that lives in the nucleus, is that going to actually become part of our genome as well or not? Would it become integrated or not? They said we do not know, and there need to be more studies to figure that out. And finally, they said it is possible that this expression of the spike protein antigen on the surface of the liver cells, again in vitro, could cause autoimmune disease. And they said, in fact, there have been studies that have shown that after vaccination, there can be autoimmune hepatitis. This is it. This is a summary. This is the concerns. This is what the study is. It is an accepted and published study. This is not a fluke. This is not a preprint. So here we are. Now I'm going to go to the second part, second level of depth for this one. And that is, so if you just wanted to hear this much at what the study is and where are the concerns and what is the mechanisms, we're done. Now I'm going to go over the main structure of the study without materials and methods. So I would not go in deep part of those. That is a third level depth, which we can do at a different time. So here is what the authors are saying. They say that we know there are studies previously done that show, for example, there are animal studies that show that if you put lipid nanoparticles, vaccine, in the animals, their liver can be transiently damaged. That is, their liver can be temporarily inflamed. They recover, but for some time they become inflamed. I had done a discussion in 2020 when I discussed the Moderna for the first time. I discussed the accumulation of the lipid nanoparticles in the liver. So here is one study. The point they're making is that here we have some precedence of these kind of observation. The other study that they have referred tons of studies, but the ones that they're using, the second study they said that we know that SARS-CoV-2, now we're not talking about the vaccine, we're talking about the SARS-CoV-2 itself. They said that there are studies that show that the RNA of the SARS-CoV-2 in some in vitro studies have been shown to become reverse transcribed. Reverse transcribed, whenever you hear that word today, that means from RNA to DNA. Reverse transcribed and become integrated in the DNA, become integrated, become part of that DNA. Now, would that cell survive or not? Would that be killed or not? That's a different discussion. But they say in vitro studies have shown that with the virus itself. And they said if that is possible with the virus, maybe it is happening with the vaccines as well. And that is what they wanted to answer the question. So here is what they did. This particular study, what they did was they took hepatoma cell, liver hepatoma cells. Liver hepatoma is a tumor. This was a Japanese person, 57 years of age. I believe in 1982, he donated these cells from his liver hepatoma. The quality of these cells, these are called HUH7, human hepatoma 7 cells. The quality is that they are dividing cells, they are cancer cells, so they divide. So if you wanted to see the dynamics of something in dividing cells, then you can use this cell line to do that experiment. And so here, The authors took this cell line, they put Pfizer-BionTech vaccine on it in various quantities, 0.5 milligram, I believe, microgram per milliliter, 1 microgram per milliliter, and 2 microgram per milliliter. They said that is actually a very small quantity because at the injection site where we have given 3 microgram in uh, zero point 0.3 milliliter or something. So you can see the math they've done. And they said it comes to be about 100 microgram per milliliter concentration of the actual dose of the vaccine. And they only tried 0.5, 1, and 2 microgram per milliliter. So the cells are there, vaccine is placed on them. Of course, now the question is, will the cells take up this vaccine? So that was the first thing. Nobody had proved that yet. And they said within 6 hours, not only the lipid nanoparticles were picked up by the liver cells, but liver cells actually within 6 hours created the DNA. It was such a fast process. So what happened? Once they put the liver cells and the vaccine together, vaccine entered the liver cell. Then they broke the cells, the liver cells, and they took out the material from the liver. They took from these cells, they took out the RNA from the cells, they took out the DNA from the cells, and then they saw various kinds of RNA that they were interested in. And again, I'm leaving that part, that how did they do it, what were the markers and primers they used. That's a third level depth. I'll leave it. So for the time being, just this, that they put the vaccine in the cells, they let it work there for 5-6 hours, then they broke the cells, took the material out and analyzed it. When they analyzed it, how did they analyze it? They took those genetic materials and increased them by PCR amplification. So we are all very aware of PCR. You give in the PCR machine, you give them some genetic material, and the machine would just keep making double copies with each cycle, and it would just double it up and make a pile of the genetic material. Now you can easily assess various things in that genetic material. You have a pile of things. So they said we created the genetic materials pile from these cells, and they said we were surprised to see that there was a huge number of messenger RNA in there. They didn't use the word surprise, but they said there was lots of messenger RNA. Good. This is the point of the vaccine. It goes into the cell, it makes more messenger RNA. Fine. So far, so good. But they said what we also saw. So now this is a nucleus. Imagine around the nucleus is a cell. The nucleus itself is this closed thing. I have removed the cell and I have opened up the nucleus for your kind viewing. And here is the DNA, our regular cell's DNA. And what they found was very interesting, that a special gene in the DNA was overexpressed. It had become very excited. It was making copies of itself and making mRNAs. And that gene was long interspersed nuclear element 1 or line 1 gene. Why the heck did line 1 gene become increased? Well, there are many genes that can start becoming increased when you add a foreign material in a cell, and especially when you cause viral infection type things. So there were some other genes that should have increased as well. These are all collectively are called housekeeping genes. And they found out that the other housekeeping genes were not increased, but this gene was increased. Very interesting. Now, when this gene is increased, then what would happen? This gene is called endogenous reverse transcriptase. This is a very unique genome. About 17% of our genome is made up of this structure, and it is endogenous. That means found within us. Reverse transcriptase means it can take a piece of RNA and convert that into a DNA. So vaccine's presence upticked the gene that will make a machine, a protein, which will increase the conversion of RNA to DNA. Look at the setup. This is happening all by itself inside the cell. So the gene is now expressing. When the gene would express, what does the gene expression mean? Inside the nucleus, when the gene expresses, we make mRNA from it, messenger RNA from it. We make copy of this recipe, and that copy comes out of the DNA and goes in the cell cytoplasm. So here now I've closed the DNA. So this is the closed DNA. These are some mitochondria. These are Golgi apparatus, other things there. But from the nuclear pores, the holes in the nuclear envelope, are these RNAs coming out. There are many tons of kinds of RNAs, but we are concerned with the RNA that is line 1 RNA. When that RNA comes out, what does it do? It goes to ribosome. Our ribosome's function is to take the RNA or the recipe and then cook it, make protein from it. So the ribosome, they saw, had started making the line 1 protein. Remember, the line 1 gene was expressing, creating line 1 messenger RNA or the copy of the recipe. Then that messenger RNA came out in the cytoplasm and went to the ribosome. Ribosome is a chef who reads that recipe and makes that protein. So you read the chicken tikka masala and you make the chicken tikka masala, or you read the whatever vegetarian thing and you make that vegetarian thing. That is what is happening here. So it makes line 1 protein. Now they saw something interesting. In the line 1 protein, there is a part of the protein called open reading frame 1. You must have heard the open reading frame 1 with the virus as well. So many DNA materials or RNA materials have various kind of reading frames. So there is an ORF1 of the line 1 protein. That little piece of protein, I hope you followed this much. In the DNA, line 1 gene expression increased. That made line 1 gene mRNA, which is the recipe, copy of the recipe. That recipe comes out in the cell cytoplasm. Inside the cell cytoplasm, the chef, the ribosome, picks that recipe up and makes and cooks that thing. That thing is called line 1 protein. In the line 1 protein, in this little food you have cooked, there is a part in there which is called open reading frame. This part of this protein actually goes back in the nucleus. It is made outside. Everything is made outside of the nucleus. Then it went back in the nucleus. What did it do inside the nucleus? When it went back, over there it found the RNA of the BioNTech vaccine and started converting that into started doing reverse transcription to convert that into DNA. So they found a bunch of DNA present inside the nucleus. Now we are looking at the third study. There are tons of other studies. Remember, there was a study where we said that spike protein goes to the nucleus, co-locates to the nucleus. It may actually not just be the spike protein, but this genetic material, maybe. Or maybe that happens plus this happens. So that study said spike protein co-locates. This study is showing that the vaccine's RNA goes to nucleus or pulled into the nucleus. Over there, line 1 reverse transcribes it into the complementary DNA or the DNA that is unique for the BioNTech vaccine. So RNA is now converted into DNA. Now, this part is open, that what will it do? When will it get degraded? It will get degraded. Remember that adenovirus-based vaccines do that too. They actually inject the DNA in the nucleus. That gets to do its function and then it is degraded. So maybe it is degraded. Maybe it is not degraded. Maybe it is DNA toxic, genomic toxic, or genome toxic. Maybe it is not. That is what they did not talk about. They said, these are our curiosities, next studies need to be done. We have proven for the first time that the DNA is formed and it is in the nucleus. Okay, so they said within 6 hours that happened. They said that liver expression and distribution of line 1 increased. When that increased, the reverse transcription function increased. When that increased, there were DNA produced. Was it integrated? They did not know. Then they said, would this vaccine go to placenta, and would this same thing happen somewhere else as well, or maybe in the baby? They do not know. Also, spike is expressed, which can create an autoimmune issue. That is the discussion. I haven't gone to one more deeper level, and that is to actually pick up all of these materials and discuss how these charts were showing the presence of various pieces in the DNA and RNA. If you are interested, I'm saying in the DNA and RNA, I meant in the nucleus and cytoplasm. If you're interested in seeing that part, that how did they do it, please let me know and I would do another study, another video for that. So here is the discussion. Um, First of all, please, there is a link in the description of this video. That link has lifetime access link to Dr. Bean, where we have like 1,000 more videos. And those videos, each one of them should be $200. But you would see that that link is so inexpensive. As I said it yesterday, it's like a gift. So use that link if you would like to support this work, plus become part of Dr. Bean and keep seeing new work that we are doing and the old work. That's one. Number two, if you'd like to support this work, there are links also in the description for buying me a coffee or using PayPal, or you could also become a patron. So that is the basic discussion. And the easiest thing also is to like, subscribe, and share as well. Now, let me just very quickly see, without trying to make this video very long, I just want to see the questions. If there are more questions, we can answer them tomorrow. I just don't want to make this video long. So Priya says, we eat so many animal food and never seen any mRNA reverse transcribed and added into our DNA. Why only SARS-CoV-2? Is the viral RNA different? No, so they did not say that it is integrated. So unless somebody says, we cannot assume that. So Rima says, I'm not sure what this means. How could Pfizer not know this? So Pfizer may know this. According to the authors, this is the first time they have shown it for the liver cells. Now, they may have seen it for others, they may not have seen it, don't know. But because this type of a study and observation was not present before, then this is the first claim of showing it. And it is an accepted study. So that means they cannot just make up something and put it in a preprint and then let the news cycle run. This is a accepted and published study. So what they're claiming here must be accurate. Okay, so this is the discussion. One more question and then we stop. Otherwise, the videos become long and nobody watches them. Radmila says, does the same happen with the infection or only with the vaccine? So remember, I discussed in the very beginning of this study that they have given reference of a study where SARS-CoV-2 itself, the virus, also has its RNA reverse transcribed and present in the nucleus. So it can happen with at least according to the studies, that was also an in vitro study. This can happen with the virus as well. So Texas says they found the mRNA after poorly washing HEK7 cells and then immunofluorescently tagging for the mRNA. Not sure the methodology, assume mRNA in the nucleus only. So this study, are you talking about this one or some other? In this study, they're saying we separated the DNA and then we made sure that DNA came from the nucleus and not from outside. And then they also made sure that the DNA is a reverse transcribed DNA of the RNA for the BioNTech. So they took care of all of those things. That is it in the nucleus versus cytoplasm? Is it really the DNA for the transcribed vaccine or not? But if you're talking about something else, and I'm sorry. So this is where we are at. Thank you very much. Once again, please like, subscribe, and share. And there are links. Is that why IVM helps? That could be one reason as well. IVM is just such a versatile thing in all of these issues. Dr. Bean, do you think that was done on purpose, any purpose, or they didn't expect it happening? This is the first time that they're showing it. Now, if let's say Pfizer knew it or BioNTech knew, I doubt it, but let's say if they knew it, then they didn't tell us. There is no study. According to the author, this is the first time they're showing this happening in the liver cells. So again, without knowing exactly who was at fault or who was doing what, it is not good, because as soon as I say, let's say I make a claim here that they knew it. Now I have not seen that they knew it or not. Then all of a sudden the whole discussion falls apart because one of the premise in there is wrong. So I do not know the answer to that. Cool. So thank you very much. Duke says what time zone Dr. Bean is in. I am in Pacific time zone. It is about 10 o'clock here at night. Thank you. I will see you again. There is a link in the description that offers you lifetime access to Dr. Bean at a very low price. It's a gift. All right. Bye bye. Please like, subscribe, and share.